Hello everyone. In this video I'm going to be looking at the basic structure and function of proteins. We'll talk a little bit about what proteins actually are, what they're made from, and in that we'll look a bit about amino acid structure. We'll then talk about the different levels of organisation in terms of protein structure, because it starts off quite simple with just a chain of amino acids and then builds up into a bigger, more complex quaternary structure. We'll then look at some of the functions of proteins and then finish just by mentioning briefly what's called the Biuret test for proteins. So given an unknown sample, how we could prove or test for the presence of proteins there. So let's start with what they are first of all. They are an essential part of a healthy diet. We talk about the 7 food groups of carbohydrates, fat, vitamin, minerals, water, fibre and protein. They're a key component of a healthy diet, and they're made of amino acids which the body uses to make new proteins for a huge variety of functions. So if we just think just of some functions that you might be aware of, they're used structurally in muscles, in collagen, in keratin, for example. Enzymes are proteins, so they're used in metabolism. They're used in carrier molecules like hemoglobin, albumin. They're used for antibodies. Antibodies are proteins. Got hormones like growth hormone, estrogen, progesterone. They can be used in membrane pumps. So we're thinking ATP pumps for receptors, neurotransmitters, and for movement. So we think about actin and myosin filaments. So there's a great deal of uses and functions of proteins. So let's think about what they're made from. The basic units, or monomers, as they're referred to, are, as we said, amino acids. So I think that's where we need to begin. So the basic, if we just put here, basic unit is the amino acid. And you can see on the left, I've used this picture because it's quite nicely shows the sequence that really you have in terms of protein structure, how you can start very simple at the top and then build up to a quaternary structure a lot more complex at the bottom. So I'll talk about those when we get to them. So the basic units of proteins are the amino acids. Now, an amino acids tend to have the same general structure. They've got a carboxyl group. So if we just draw this down here, they have a carboxyl group, which is COOH, just keep it very simple, and an amino group, which is an NH2. So we'll just put H2N there, put a little H there, and that's attached to the central carbon atom. But the difference between amino acids is what's called the variable group. Now this is often given by an R. So that R refers to the variable group. And that's what gives rise to all these different amino acids. Now I think the body makes naturally about 20 of them. And you can see on the picture on the left, the little circles, red, green, yellow, orange, they've got 3 letters in. So you can see ASN, GLY, PHE, GLU. They're short hand terms for the amino acids. So like ALA, for example, is alanine, ARG, arginine. So you've got all these different amino acids. Now, a dipeptide is formed when 2 amino acids join together. So you have one amino acid, if we just draw it as a blue circle here, one amino acid, that is a single monomer. When we have 2 of those joined together, we form a dipeptide. Now more of those together, so let's just put in, let's say, 5 in this sequence. Many of those form what's referred to as a polypeptide. And that's essential for what I'm about to describe shortly. So proteins are made up of 1 or more polypeptides. So you said they're primarily made from amino acids. And amino acids are linked together, just as a bit of an FYI, they're linked together by what's called a condensation reaction. So we'll add that there, just because it's a bit of an interesting thing to note. So they're formed by what are called condensation reactions. So let's go back to our initial protein structure. Proteins are big, complicated molecules. So they're much easier to explain if you look at it in terms of these 4 levels. So it starts with the primary structure, the amino acid sequence. I'll put a little number 1 at the top of this picture here, because that's where we're starting here. Now that's a sequence of amino acids determined by DNA, and it can range from about 50 to 1,000 amino acids. So it's the sequence of a chain of amino acids. We then move to our secondary protein structure. Now that occurs when the sequence of amino acids are linked by hydrogen bonds. So you get hydrogen bonds in the main chain between the amino acids, and this makes it automatically coil into either an alpha helix or fold into what's called the beta pleated sheet. So that's why we can see we've got the alpha helix here and the beta sheet there, and there are secondary structures. Now, if the coiled or folded chain of amino acids is folded even further, we then get our tertiary structure. So our tertiary protein structure occurs when certain attractions are present between these alpha helices and pleated sheets. So when we get attractions between the alpha helix and the beta pleated sheets, attractions start to form. So it's almost as if I do this in red, if I just put a line here, that's meant to represent an attraction between the alpha helix and the beta sheet. So when those come together, that's when you get this tertiary structure. The polypeptide folds up to give a 3D shape. And then finally we get the quaternary protein structure at the bottom. So we had 1, 2, 3, and then finally we've got our quaternary structure down at the bottom. Now that's a protein consisting of more than 1 amino acid chain. So it's essentially the linking of 2 or more polypeptides. Now just as an example, insulin is made of 2 chains, collagen is made of 3, and hemoglobin actually has 4 chains. And that's the one I've used in this example. In the bottom left you can see it's got a sort of visual representation of hemoglobin, and we've got the 4 chains here. There's A, B, C, and D there. We've got these 4 polypeptide chains that have come together to form this quaternary structure. So let's think now about what actually is it that holds the folds of a protein together. So we're thinking here about the bonding essentially. So let's make a little side note here. Bonding, because I think this is key to mention. So we've got hydrogen bonds, which we've referred to already. So we've got hydrogen bonds there. We've also got what are called disulfide bonds. Now, as a third thing, we have what's called an ion bridge, and I'll expand on that in just a moment. So we've got the ion bridge, and what that is an attraction of a positive R group and a negative R group. So I said on the amino acids that you have this R group, this variable group. Some would have a positive charge, some may have a negative charge, and when they interact, when this chain folds up. So if you imagine you've got this chain of amino acids, some of the R groups are positive, some negative, and then you fold this chain up, you can see that some of those positive and negative groups are going to interact, and that would create an ion bridge, just a strong bond to keep these folds or folded chains together. But then you've got, I'll put number 4 here, hydrophobic interactions. We'll just put that there, and I'll expand on that in a moment. So hydrophobic interactions. Now, they're bonds between hydrophobic R groups. Now, what I want to do is just quickly explain the importance of polar and non-polar amino acids. When we say polar, I'm referring to hydrophilic or water-loving amino acids, and non-polar hydrophobic, water-hating amino acids. Now, this is important because the arrangement of these particular amino acid residues will do 3 things. They will, 1, control the position of proteins in membranes. They'll, 2, create hydrophilic channels through membranes. And 3, they will be involved in the specificity of active sites in enzymes. So I've done a video talking about enzyme specificity, saying that the active site has a very particular shape, and that in some part is down to the arrangement of these polar and non-polar amino acids. So now we've mentioned that, I want to go into more about the types of proteins that we've got and link to that the function by giving some particular examples, and then we'll look at the Biuret test. So for this, I'm going to shrink the screen just a little bit there, and we'll look at some of the types. Now, when we think of proteins at A level, we think of them in terms of 2 types. Type 1 that I teach my students about are the fibrous proteins, and then the second type are those that are globular. So let's think what we mean by these. The fibrous proteins, they're the ones that have an elongated shape. I'm just going to make a few little notes here. They're the ones that have an elongated shape, and they tend to be insoluble in water. Now, examples that I can think of include, so I'll make a note of these as well, e.g., so the ones that I'm going to refer to are ones like collagen, got keratin, skin, myosin, we think about muscle, and I'll put that elastin too. So let's just think about those. Collagen, if we take that one, that is a structural protein that strengthens bone, tendons and skin. It consists of 3 long polypeptide chains lying parallel to each other with cross links between them. If we think about myosin, that works with another protein, actin, to contract muscle fibres and so to cause movement. And I've talked about that in another video that I've done. So they're an example of fibrous proteins. We can see there for muscle and for structure. Let's think about globular proteins. Because globular proteins, unlike the fibrous ones that have a more elongated shape, these are ones that are more compact and have a more rounded shape. So we put compact and rounded shape there. And these ones tend to be water soluble. Now, examples that I can think of here are hemoglobin and immunoglobulins that I referred to at the beginning. These are globular proteins. Hemoglobin binds to oxygen in the lungs and transported to the tissues, and immunoglobulins are antibodies involved in the defense against disease. Now, in a separate video I've talked about antibody structure in detail, and I've said how antibodies are made up of 2 heavy chains, which are long, and 2 light chains, which are basically short polypeptide chains, with what are called variable regions. And as you might guess, they're regions where the amino acids differ. So you've got an example there of 2 fibrous and 2 globular. Fibrous ones, collagen and myosin, globular, hemoglobin and immunoglobulins. So I've got a little bit there about the function of proteins, and in the video I've talked about the basic structure, going from the primary structure of the amino acid sequence into the alpha helix and beta sheet of the secondary structure, further folding to give the tertiary, and then finally we've got the quaternary structure where we've got a complex of protein molecules. Now, one key thing that's really important to say at this point is that not all proteins have a quaternary structure. Some are made of only 1 polypeptide chain. So you get many that are just simply at the tertiary level. So I did say that I would finish up by talking about the Biuret test. It's a very quick test for proteins. So if you need to find out if a substance, the random food sample, has any protein in it, you would do this test. And it's got 2 main stages. The solution to be tested needs to be alkaline. So first you've got to add a few drops of what's called sodium hydroxide solution. Then you add some copper 2 sulfate solution. Now, if the protein is present, the solution turns purple. If there's no protein at all, it will stay blue. The colours can be fairly pale, so it's important to maybe get a second opinion on this. So you you take the test solution, you add sodium hydroxide and then copper 2 sulfate solution. If it stays blue, then it's a negative result, and if it goes purple, it's positive. But I talk more about in-depth biochemical tests in another video. So there we have a little bit about the basic structure of proteins and a little bit about their functions. Okay, hope all that helps.